Can you hear me? We'll start the class now. So in today's class, we will look at all the questions in relevance to what was taught in week 8 of the lecture for NPTEL's Pharmacognosy and Metabolic Engineering. So the first question is, higher molecules of prenyl phosphates are formed by head-to-tail addition of isoprene units. Identify the enzymes leading to the formation of geranyl pyrophosphate from isopentenyl pyrophosphate. 4 different options are given here: isoprene synthase and prenyl transferase, isomerase and prenyl transferases, prenyl transferase, and option D is isoprene synthase and isomerases. You can take a couple of minutes and then think about the question, and then we'll come back to the answer. Okay, so here we are talking about how isoprene units get attached, which would need a transferase enzyme. So which of these do you think is the right answer? If you look at the pathway, okay, this starts from the precursor. What we are looking at is IPP, which is isopentenyl pyrophosphate, that gets converted into dimethyl allyl pyrophosphate. And prenyl transferases can catalyze the sequential head-to-tail condensation of isoprene units. So here When you have geranyl pyrophosphate, it is synthesized from this DMAPP and IPP by this transferase enzyme. And farnesyl pyrophosphate is formed by adding one more of this IPP to this GPP. Then this geranyl pyrophosphate is synthesized by adding one more of this IPP to this FPP. So we have both an isomerase and a transferase. The isomerase is needed for converting IPP to DMAPP, whereas transferase is needed for chain elongation. So option B is the right answer. Right. We will go to the next question. The next question is, match the monoterpenoids from column A with their cyclic nature in column B. And you have myrisine, menthol, limonene and linalool. A few of these are cyclic and a few of these are acyclic. So you can think about this question for a couple of minutes. Think about their structure, and then we'll get back to which of these are cyclic and which of these are acyclic. Okay, so if you look at this, the next slide, I have given the structures of all these four compounds. The first one towards the extreme left is myrisine, the second one is menthol, the third one is limonene, and the fourth one is linalool. So if you look at this, menthol and limonene are the cyclic compounds here. So in column A, 2 and 3 belong to one of column B, which is there in option D. So menthol and limonene are cyclic, myrcene and linalool are acyclic. Right. We'll go to the next question. Okay, the next question is, cyclic monoterpenoids are formed from their linear isoprene molecules via cyclization. During the reaction, which stable or unstable ion intermediates are formed that undergo cyclization and further transformation? And 4 options are given here: carbocations, carbanions, carbohydride ions, and hydroxyl ions. You can think about this question as to how the cyclization happens and what is the kind of ion that is needed for cyclization. So to answer this, just think about the precursor moiety, whether it has a positive charge or a negative charge. That will be easier for you to decide what kind of ion it would be. One clear way to think is all of these are carbon related molecules, so it can't be hydroxyl ions. So think about this and then we'll get back to the answer. Okay, so we looked at the previous questions which had discussed on what is the kind of enzymes needed, whether it was an isomerase or a transferase. So there we had this geranyl pyrophosphate. Now this is the linear precursor. When this geranyl pyrophosphate, it undergoes ionization, it can form a carbocationic intermediate. These are very reactive and that is what gets cyclized to form cyclic monoterpenoid structures. You also have a few enzymes like terpene cyclases that can help in the cyclization process by stabilizing the carbocation. And I have an image taken from a taken from a paper where they show what is the kind of carbocation that's formed. It gives you the electrostatic surface potential for the side chain aromatic groups of phenylalanine, tyrosine, and tryptophan. And here also, if you see, they show how this enzyme FPP synthase catalyzes IPP molecule to the chain, and how DMAPP and IPP would combine to form GPP. This is the reaction that we saw in the first question. Right. The next question is, identify the compound from the following list that is not a product of cineol synthase. Option A, limonene. Option B, sabinine. Option C, linolol. And option D, beta-pinene. Okay, the question was about cineol synthase and which of the products it could produce. So this enzyme, cineol synthase, it converts GPP, which is geranyl pyrophosphate, into 1,8-cineol, which is colloquially called as eucalyptol. But because of this promiscuity, it could also produce limonene, sabinine, and beta-pinene. But linolol is not a product of cineol synthase. And linolol is being produced by this enzyme called linolol synthase. Right. The next question is, For metabolic engineering purposes, one Arabidopsis thaliana transgenic line was developed that expresses this Clarkia brewery linalool synthase gene. The rationale behind this work was that the substrate for linalool synthase (LIS) enzyme would be available in Arabidopsis cell and could produce linalool. This linalool would get released from Arabidopsis flower as floral scent. However, this ambitious experiment did not yield expected results because of one of the following constraints. Identify the correct answer. Option A, linalool did not form at all in the target plant. Option B, linalool got readily transported to roots. Option C, linalool got trapped in vacuole in the form of linalool glycopyranoside. Option D, LIS is a bidirectional enzyme and the Km for backward reaction is much higher than the Km for forward reaction. Hence, no linalool accumulated in the target tissue. You can take a couple of minutes, think about the question, and then we'll discuss what this enzyme is about, what is the substrate, and what happened in this system in Arabidopsis. Okay, we will look at the options one by one. First, did linalool get formed when will linalool not get formed? When the linalool synthase gene doesn't have a substrate, linalool will not form. Because Arabidopsis thaliana has the substrate for linalool synthase gene, of course, linalool formed. So, option A, that linalool did not form, is wrong. The second option, okay, we look at the answer here. They say that in these plants, these volatile terpenoids are majorly glycosylated. Glycosylated means it has a glucose moiety added to it. This is done because if it is stored, if it has to be stored for long term purposes, it cannot be in the volatile form, and adding such a glycoside would prevent it from being volatile. And in Arabidopsis thaliana, this was observed. In Arabidopsis thaliana, this was observed to be stored in the vacuoles, which is why it could not produce the expected floral scent that is associated with linalool. Okay, for this question, you need to first think which of these is the volatile resin and which is the non volatile. Is turpentine volatile or non volatile? And what is turpentine? So, turpentine, it is volatile. It is produced from terpenoids, whatever we were discussing earlier. It has a composition that includes alpha pinene, beta pinene. Beta pinene is what we saw in an earlier question. It also has limonene. So it's a mixture of multiple monoterpenes. Okay. Now the question is, oleoresins, these are comprised of which of these? So because we have now concluded that turpentine is volatile, it's either option C or option D. Okay, if it's difficult, maybe think about what oleoresins are. Yes, you can unmute.
Is it option D, sir? Oleoresins are the natural products from the essential oils.
Yeah, it has both the resin and the turpentine. So we were able to conclude that turpentine is made up of terpenes, like we mentioned about beta pinene and limonene. Now we just have to look at the ratio. Which of these is higher in oleoresins? Is it the turpentine that's higher or the non-volatile resin that is higher? Okay, there's a small. Oh, okay. Swati, you were saying option D. Non volatile resin is to volatile turpentine 50-50, right? So, one thing is that usually in these oleoresins, you have more of the non volatile resin and less of the volatile turpentine. So, actually, none of these options are right. If option C has 80:20, that would be right because you have a maximum of the non-volatile resin and a minimum of the volatile turpentine. So here The non-volatile turpentine, it's either a solid or a semi-solid part. And this volatile turpentine, we know that it is a monoterpene. That makes it volatile. The next question is, find out the correct statement about the secretions that occurred in Hevia brasiliensis bark. Option A, produced in phloem, accumulates in xylem vessels. Option B, produced in xylems, accumulates in laticifiers. Option C, rubber present in the latex is cyclic monoterpene in nature. Option D, rubber present in latex is polyterpene in nature. So take a couple of minutes, think about where this is being produced. We have learnt about laticifiers and its relevance to latex earlier. Think about that, and then we also have learnt about sieve elements and vacuole and what is stored where. Just think about that. That will help you eliminate the wrong option, and also whether it has the latex that's being discussed here, whether it has cyclic monoterpene or if it has polyterpene. Which with that you will be able to eliminate the wrong options here. Okay. Okay. So the Answers are this Hevia brasiliensis, it's just rubber tree. It produces latex, and latex is natural rubber. So if you look at the chemical composition of this natural rubber, it has cis 1,4 polyisoprene, which is polyterpene. That means it has many isoprene units that are covalently linked. And like we learnt in the initial classes, this production of latex occurs in specialized tissues called the laticifiers, and these are present in the phloem. There is no relevance for xylem here. Okay, and one more thing is, if we look at monoterpenes, they contain only two isoprene units. The chemical formula for monoterpene is C10H16. So with that, we'll be able to say that option C is incorrect. So if we look at the options here, option A, that latex is produced in phloem but does not accumulate in xylem vessels, instead it remains in the laticifier. The next option is latex is not produced in xylem, it is formed in phloem. That's what we saw. And third option is rubber is not cyclic monoterpene, it is long chain polyterpene, which is polyisoprene. Okay, fine. Precursor for terpenoid biosynthesis, that is isopentenyl diphosphate IPP, can be produced via MVA or MEP pathway. Identify the correct combinations of primary metabolite precursor that enters to these pathways to produce isoprene units. And 4 options are given here: acetyl-CoA, malic acid and pyruvic acid, malic acid, pyruvic acid, shikimic acid, acetyl-CoA, glyceraldehyde 3-phosphate and pyruvic acid, acetyl-CoA, glyceraldehyde 3-phosphate and malic acid. So for one of the questions, we had looked at the pathway from which it's being produced earlier. Try to recap the pathway where we looked at the effect of transferase and isomerase enzyme. So if we go to the very beginning of that pathway, we will know which of the precursors are required. You can take a couple of minutes, think about this question. We'll get back to it.
Option C, sir.
Okay, what is the rationale for it? Do you want to start by going through the pathway? Okay, we'll first look at what MVA and MEP pathway are. Do you know what these pathways are?
Mevalonate pathway and MEP from
Okay, what is the precursor for mevalonate pathway? Okay, why do we have two pathways? Any idea about that?
the precursor is IPP. No, the precursor is this one. Glyceryl phosphate and acetyl CoA.
No. So, if you look at mevalonate pathway, there it starts with two acetyl CoA molecules. Acetyl CoA plus acetyl CoA, that will give you acetoacetyl CoA. And then you have a number of steps after which it forms mevalonate, and finally it will form isopentenyl diphosphate. I'll show the image where, and this happens in the cytosol. Whereas the MEP pathway, can you recall what the full form of MEP is? Any idea? No issues. MEP means methyl erythritol phosphate, which is produced as a part of this pathway, and this happens in the plastid. For this pathway, you need glyceraldehyde 3-phosphate and pyruvate. Again, this will also produce isopentenyl diphosphate. But in addition to isopentenyl diphosphate, both of these from both of these pathways, if isopentenyl diphosphate is being produced, it will give dimethyl allyl diphosphate. But of interest to us is isopentenyl diphosphate only. So here, so the mevalonate pathway, there the precursor is acetyl CoA. Like I told you, two molecules of acetyl CoA form acetoacetyl CoA and all the way produce mevalonate and finally IPP. Whereas in methyl erythritol phosphate, which is the MEP, that occurs in plastids. Here G3P and pyruvic acid, this will give you IPP. Right. So this is the pathway here. For MEP we have it here. So if we look at the options, in option A, they have given malic acid also, which is wrong. In option B, they are saying shikimic acid. We have seen earlier that shikimic acid is involved in producing tryptophan and tyrosine, that are involved in the production of flavonoids, not terpenoids. And option D, they have again told that malic acid, but it is not needed for IPP biosynthesis. So this leaves us with the options acetyl CoA, glyceraldehyde 3-phosphate, and the third one. Sorry, what is that? The next question is, match the terpene product from column A that derives its final isoprene precursor unit. And in a column A, you have carotenoids, monoterpenes, sesquiterpenes, and cytokinins. And in column B, you have geranyl diphosphate, farnesyl diphosphate, dimethyl allyl diphosphate, and geranyl diphosphate. Take a couple of minutes, think about this, and then we'll get back to the question. Okay, to answer this question, you can think about what cytokinins are. You might have come across this word cytokinins in immunology. Okay, let's generally look at what these are. So first, terpenes, they are classified based on the number of isoprene units. One isoprene has one C5 unit. Okay. If we look at carotenoids, these are having 40 carbon units, which means that these are tetraterpenes. One example is this geranyl diphosphate, which has a C20. Now carotenoids like beta carotene and lycopene, these are derived from geranyl diphosphate, which is the precursor for diterpenes and tetraterpenes. And if we look at monoterpenes, which is C10, it has geranyl diphosphate. Again, this will give us this linalool, limonene and menthol. Whereas sesquiterpenes are between your monoterpenes and tetraterpenes. It has C15. And an example of that is farnesyl diphosphate. Finally, cytokinins. Sorry, I made a mistake confusing cytokinins and cytokines. Cytokinins are plant hormones that can regulate the levels of these. So, here, dimethyl allyl diphosphate, DMAPP, this is a part of that. And these are adenine based molecules. Right. The next question is, identify the plastidial pathway operating in plants for biosynthesis of terpenoids. So by plastidial, what we mean is the one that is happening in plastids. And 4 options are given here: shikimic acid pathway, mevalonic acid pathway, methyl erythritol phosphate pathway, and pentose phosphate pathway. So here, earlier we discussed about 2 pathways that give rise to your IPP. One happens in the cytosol, one happens in the plastid. Think about it, and the pathway that happens in the plastid is the answer for this question.
The question on the screen is the carotenoid monoterpene cis-bitter. This one is option D, no? Okay.
1 minute. Wait, the question hasn't changed, is it? 1 minute. Right. So, like we looked at this earlier, this mevalonic acid pathway, it happens in cytosol. And methyl erythritol phosphate pathway happens in plastin. So, these are the questions that we have for this week. If you have any other doubts, I'll stay on the call. You can ask me.
Can you show the first question?
Yeah, sure.
At least.
Sure.